talk to you a little bit about community phylogenetic structure and how we think about it and how we analyze it. So specifically, drawing on evolutionary history in terms of phylogenetic signal to understand how species respond to environmental gradients, while simultaneously taking into account the fact that when we actually go out into the field and look for species, we don't necessarily always see them even if they're there. So that's the imperfect detection bit. So in the past 2 decades or so, we've made tremendous strides in how we analyze data in both evolutionary biology and in ecology. And in evolutionary biology, a lot of this is taken the form of comparative methods. So the recognition that species are not necessarily independent data points, and that we can actually use that non-independence to really understand how traits evolve through evolutionary time. In terms of ecology, a large approach in the last 10 years or so has been thinking about this problem of imperfect detection, and methods in terms of hierarchical models have been developed to try to measure that imperfect detection and account for it statistically. So I'm going to be talking about the interface between these 2 fields and how we can leverage both of them to come towards a better analysis of community data. But before I dive into that, I just want to talk to you a little bit about environmental gradients. So this is a focus of a lot of modern day community ecology to understand why species exist where they do. Often we have to think about the underlying environmental gradients that exist in the world. So I personally study deforestation in tropical regions, specifically in Costa Rica. So if we think about communities of species in these environments, there are tons of species of, say, birds that are restricted to tropical forests. But there are also tons of them that are restricted, somewhat bizarrely, into these new evolutionarily novel deforested environments. So we might want to understand why that is. And one way to begin to approach this question is through community phylogenetics. So we can think about what are the consequences of deforestation for, say, avian phylogenetic community structure. And kind of the canonical way to go about doing this is to take your favorite bird phylogeny and then think about how closely related those individual species are in communities along the deforestation gradient from completely forested sites to completely deforested sites. And when you go out into the field and you collect this data, what you see is that as you move from forested to deforested environments, species, in terms of their mean phylogenetic distance from one another, tend to get more and more closely related. And the canonical explanation of why this occurs is that there is some environmental filtering occurring in deforested habitats. That is to say, there are some evolutionarily conserved traits that these birds are expressing that somehow allow them to survive in deforested environments, so that only those species with those phylogenetically conserved traits can actually manage. But what if we want to actually dive a little bit deeper and think about what individual species are doing? So I'm a shameless reductionist, so I really want to understand what these individual species are doing. And particularly, how important is phylogeny per se in mediating that response to the environmental gradient, say deforestation or anything else that you might be dealing with. So thinking about these types of questions is super easy when you're dealing with something like body size that's directly observable from the animals. You say, go out, collect some salamanders, measure how much they weigh, and bam, you can understand the evolutionary history of how that trait evolved in a very nice direct way. But when you're dealing with something like a response to environmental gradient, it's a little bit more tricky because that response has to be inferred from a lot of additional data. What I'm doing here is building off of this beautiful framework that was published a few years ago by Ives and Helmus that leveraged these tools for generalized linear mixed effect models to analyze community data. But when you go out into the field, you quickly realize that you have a problem in that detection screws you. This butterfly is way easier to detect than is this butterfly. This frog is way easier to detect than this frog. It's like you can stare at this guy for a long time and not see it. And that's only kind of the very surface level of the problem with imperfect detection. Because detection can itself depend on environment. So this cute little pica is super easy to see against a green background. But when it's in this talus slope, you might stare at it for a while and not necessarily see it. And this can result in pathological conclusions from the biological perspective. So you might conclude that picas hate these talus slopes, when in fact they're just harder to detect. And when you're dealing with single species, you can usually target your collection methods, your sampling methods, to be somewhat impervious to detection bias. But when you're actually dealing with entire communities, this isn't always so easy. so it helps to take a somewhat statistical approach and try to at least account for this fact. And the way that we do this slide will hopefully load sometime. There we go. The way that we can do this is what I've been calling the phylogenetic occupancy model. So this is a way to take your raw data, your detection history of species at a bunch of sites along some environmental gradient, combine it with a phylogeny to really understand how that phylogeny governs responses to an environmental gradient. So I'm going to walk through this kind of step by step, starting over here on the right and work my way towards the left. So thinking first about the raw data, there's a lag. What you need is a detection history of some number of species across some number of sites along an environmental gradient. So in each of these sites, you visit it multiple times, and this is used to infer detection probability. So in terms of the schematic, if you say visit this site, this light color indicates that you saw it maybe 0 out of 3 times. These darker colors maybe represent 1 or 2 observations of the 3 times, and this darkest is 3 out of 3 observations. You also obviously need a phylogeny if you want to know anything about the phylogenetic structure. But in terms of the kind of underarching hierarchical model, we use these detection data to try to understand how the detection probability on a species-by-species and site-by-site basis leads to the observed data. So if we focus first on just the detection component, we have this Y, which represents the raw data of a species, site, and visit. So this is whether you detected a species at a site or not. So it takes values of 0 or 1. And it comes about due to this Bernoulli process of whether a species was truly at that site or not, again, 0s and 1s, and the detection probability of that species at that site. And this is something that will be measured from the data, parameters will be estimated from the data. So obviously if a species doesn't exist at a site, this is 0, you're never going to detect it no matter what the detection probability is. If it does exist at the site, however, whether you detect it or not is going to be entirely determined by the detection probability. So then if we move down to the occupancy component, we have the true occupancy state, which again is determined by this Bernoulli process, which is just the occupancy probability of the species at that site, which in kind of the framework of the generalized linear mixed effect model is itself determined by some occupancy intercept, which is just the occupancy probability of a given species at the midpoint of the environmental gradient, and the species' response to the environment. And this is kind of where the money is in terms of this an analytical framework, because these values, we're going to say, are correlated to one another according to the phylogeny at hand. So say, display the species, through this entire process, we come to understand that there is strong phylogenetic signal in this data set, so all these guys are responding to this environmental gradient in a similar way. They all have a positive response going from being very uncommon in this green zone to very common in the yellow zone. And this clade has the opposite trend. So, kind of now that we've established this framework, the next question is, does it actually work? Can we use the phylogenetic occupancy model to detect phylogenetic signal when it does exist? So I conducted a number of simulated detection history analyses to actually check this, and the short answer is that yes, you can. So here we're using lambdas and Hagel's lambda. It's just a way to quantify the phylogenetic signal. There are tons of different ways to think about this. This is my preferred method because it's relatively simple, but it can be made as complex as anyone wants it to be. And basically the way that lambda works, as many of you are probably familiar with, is that when it equals 1, the trait evolved entirely according to the Brownian motion expectation. So this is just inheritance from the previous ancestor. When lambda equals 0, the phylogeny offers no predictive power in terms of what species are doing, in this case in terms of their responses to the environmental gradient. So what you see here, as long as you have 32 or 64 species, the occupancy model does a pretty good job of recapitulating the lambda value that you simulated, which is good. And this is with a mean detection probability that is set at only 25% and three site visits. So this is good. But what can we actually use this model for in terms of thinking about real data? So returning to these birds in Costa Rica, running the phylogenetic occupancy model, we can come away with a more species-specific understanding of how these guys are responding to deforestation. So we have a number of clades of birds here. The agricultural affiliated ones, the deforestation affiliated ones are on the left, forest affiliated ones are on the right, and this is the response to tree cover. It's that psi beta parameter that governs the response. So you see a large number are forest affiliated, a large number are agricultural affiliated. There don't seem to be too many generalists. But the key point here is that phylogeny does provide a fair amount of predictive power here. So Brownian motion evolution explains roughly a little bit over 1/2 of the variance in deforestation tolerance. And I didn't mention this earlier, but one of the nice things about this approach is that this response to the environmental gradient is actually biologically meaningful in that it expresses the log odds of finding a species at one end of the gradient or another. So it's actually not just a parameter kind of floating in space, it actually does mean something. So in conclusion here, I view this approach as really complementary to the mean phylogenetic distance or phylogenetic species variability approaches that are typically used in community phylogenetic methods. And the key point, or at least one of them, is that it does incorporate imperfect detection. Additionally, you can detect phylogenetic conservation to response to the environment. And something that I didn't talk about here, but is extremely important, is that it's fairly trivial to incorporate traits, to ask whether traits predict whether a bird species, say, is deforestation tolerant or not, while simultaneously accounting for phylogenetic non-independence of data, which is not something that we can easily do before. And finally, this is generalizable beyond monotonic responses. So here I was just dealing with deforestation, birds either like trees or they don't. But you can also think about other climate gradients, say temperature or precipitation, where you have kind of a curve in the response. And it could be reparameterized fairly easily to think about species optimal climate space, and whether that optimum is itself phylogenetically conserved through the phylogeny. So this might be a way to kind of think about multi-species distribution models with phylogenetic effects. So with that, thank you all for coming. I'd like to thank my funding sources, and if you have any questions or data sets that you might be interested in using this approach for, please email me. So thanks a lot. Yeah, I'm back.
If the detection probability varies depending on the environment,
yeah.
Can you incorporate the covariance of environment and detection probability
Absolutely.
into the model?
Absolutely. Yeah. And that's a critical point, because that's the primary situation when you can come away with the totally wrong answer if you don't incorporate detection probability. And in fact, if you don't, during some simulations, you can kind of decrease the perceived amount of phylogenetic signal that you have by around half.
But you could just throw that covariance term into your model.
Which one?
Into your phylogenetic occupancy model.
Yeah, absolutely. Yeah.
In terms of usability, what sample space across the phylogeny must you input as data in order for this to work? So I'm saying, like, how many species do you need for?
So it'll depend on the exact topology. But I mean, in our simulations, once you get to around 32 species, it performs pretty well. I mean, with most phylogenetic methods, you lose power as you get fewer species, and this is no different. I mean, it's a little bit more stringent just because the model is relatively more complex. There are way more parameters than a simple phylogenetic signal calculation. But yeah, once you get around 32 species, it performs pretty well.
Thank you.
Thanks a lot.